Hi, Dr. Hyder. How are you?
Hi, how are you, Kim?
I'm good. How are you doing?
Fine, good. How's everything?
Alhamdulillah. Alhamdulillah, fine.
Fine, good.
Hopefully, this will be the last day of the conference.
Okay.
I believe you have the speakers moderators kit.
Yes.
Okay.
So everybody's online or not yet?
I think not yet, but we will start at exactly 3 p.m. So 1 minute before 3 p.m., you can start introducing Dr. Mario, which is our first speaker. Then we will give him 20 minutes. Then we'll move with Professor Ahmed Al-Khatib. Then
Luis.
20 minutes. And then Luis. By the way, just to tell you, Professor Luis Dai will have a recorded
40
video.
minutes?
Yeah, 40 minutes, but she will have a recorded video ready. So we'll not tell this to the attendees, and I'm not sure if she'll be able to attend at the same time. So any questions remaining for the session of Dr. Mario and Professor Ahmed, we'll take them after the recorded of Professor Luis.
Okay, so both of them, only Luis will be recorded, the other live.
Yes, which is, I believe, Dr. Mario is already here. Dr. Mario, how are you?
Hi, I'm good, thank you.
Hi, Dr. Mario.
Hi, how are you, Dr.
Fine.
Hassan?
Nice to be together.
Yeah, 1 minute. I'm trying to get this background which Kim sent me. Kim, how do I do that?
I would like to do the same, Kim. How
sure,
can I
I'll
do
forward to you. I'll forward to you, Dr. Hyder. Just give me 1 second.
I mean, I did put it on my Zoom, but it's not showing.
You need to go to the start video in the Zoom call, Dr. Mario, while I'm forwarding.
Yeah, and if I put, now is it clear? I will choose one. Can you see if it is clear or not? How it looks like?
No, I can see it clearly because your face has been...
Yeah, now it's not clear, isn't it?
Maybe because of your background, Dr. Hyder.
Now, what about now? The same?
No, yeah, still. I can see you properly.
So I will remove
it. No need to use it, but I will forward it to you, okay?
Okay.
Dr. Mario was not in the call anymore. Okay. You can stop it. You can click none, Dr. Hyder.
Yeah, I put general. About now?
Still, it has.
There's none. Okay.
Okay, perfect.
Okay, that's fine.
Okay. So I'll start calling all the attendees now. While
we have
Dr. Mario.
Yeah.
Right.
Hello?
Hi, Dr. Mario.
Now can you see me?
I can see you clear.
And you've got the background, right?
Yes, perfect.
Okay, good. I'm glad I came online earlier, otherwise we'd be in trouble.
Yeah. Currently, you are in Australia?
I'm in Perth, Australia. That's
Perth,
correct.
Australia.
Yeah.
How's the weather now?
The weather is very good. I mean, basically, ours is on the warmer side, more Mediterranean, and throughout the year. Yeah. So I think I have too many slides, Dr. Hyder.
You have 20 minutes you have for the presentation.
Yeah, I know.
Okay.
Yeah.
2 minutes before I will give you a notification.
Okay, thank you.
Okay. So 1 or 2 minutes before the 3 o'clock, before you start, I will start introducing you. So you'll use your 20 minutes properly.
Hi,
Okay.
good morning, good afternoon, everyone.
Hi, Prof. Ahmed. How are you?
Hi, how are you? You
Good?
okay? Yes,
Good.
yes. Hi, Dr. Haider and Dr. Mario and
Yeah.
Kim.
Hi.
How you doing? Okay.
Yeah.
Very good, thanks.
Are you in
Hi,
Taipei?
Professor Hammond.
I'm sorry I've joined you late because I've joined at 2, but I didn't see anybody, so I thought maybe it's a mistake. Starts at 3. Is there anything we need to sort of be aware of before presentations, or all in hand?
No, nothing. I think everything is fine, isn't it, Kim?
Yes, everything is fine. We're just waiting for at least 58 or 59 to.
Yeah,
Dr.
I
Haider
think
to start
58 I
his-
will start.
Excellent.
In
And,
6 minutes I will start.
Thanks. And Dr. Haider is it says 20 minutes. That includes the questions, is that correct? So we have to finish
We
in
will
less
keep the
than that?
questions to the end?
Oh, okay. So we have the whole 20 minutes. It's okay.
Yes,
'Cause I'll-
20.
Yes, yeah.
Yes.
That's
Okay.
good. Yeah. Excellent.
Good. So now I
How's the
click
weather in
on-
Taipei?
At the moment, I'm back in the UK now.
UK, okay.
Yeah, I've joined
Good.
the Teesside University. So
Yeah.
it is gloomy and rainy, as
Oh,
usual, as you
okay.
probably know.
Here
already.
today the temperature is 37, and so we
Wow.
started the summer, in fact.
Wow.
Mm.
That's in Dubai, is that correct?
Yes,
Dr.
yes,
Haider,
yes,
yeah.
yeah.
Yes. Hope you're keeping well with the situation. I think with the good weather, I think the better or less risk, I hope.
Yeah. I think everything is under control here. Most
Yes.
people, they receive the vaccine, so we are
Yes.
almost all vaccinated, which is good.
Oh, that's
Yeah.
very good.
Yeah.
Yeah.
For me and the family, we all got the second dose Sinopharm.
Excellent.
Yeah.
Which
Okay.
vaccine is used at the moment in the country?
We have Sinopharm in general, but also
Okay.
there is Pfizer and there is AstraZeneca.
Oh, excellent.
Yeah.
Yes.
Yeah.
Where are you based, Dr. Mario? Which country?
I'm in Perth in Australia.
Oh, in Australia. Excellent.
Yeah.
In Perth.
Perth,
Western
yeah.
Australia.
Western Australia, correct. So I'm only
Yes,
4 hours ahead of Dubai, and you're in the UK, so that'll be
Yes, we're in
7
Perth.
hours.
Yes.
7 hours, isn't it? Yeah.
7 or 8 hours. Yeah.
Yes.
Yeah.
I
So
hear
Tees-
that the winters has started already in where you are. Is that correct?
Me in Perth?
Yeah, the winter season. Because my colleagues I've got some friends in Tasmania and in
Tasmania is different, my friend.
It's close to New Zealand, isn't it? Tasmania is close to New Zealand.
No. Tasmania is south of Australia. It's a tiny
South of Melbourne, isn't it?
south of Melbourne. That's correct.
Yeah.
Whereas
Yeah.
New Zealand is south and to the east.
East, I see. Yeah.
Yeah. So it's further down. So
Okay, yeah.
being islands, Tasmania and New Zealand are invariably very cold.
Yes, yeah. And they are close to the South Pole.
Yeah.
Yeah.
Yeah. So ours is more kind of warm Mediterranean. If you go
And
to Queensland, it is very
Yeah.
tropical and very humid. Melbourne, you can get 4 seasons in one day. You know, yeah, a lot of the English who left the UK years ago prefer Melbourne because it's kind of cold and rainy and gloomy.
yes. It's home from home.
But that's right. But definitely
yeah.
it gets more sunshine, I think, than the UK.
But Perth is growing now, isn't it?
Perth is actually the state is 1/6 the size of Australia, so it's huge.
Yes.
But our population is only 2.4 million or something.
Which is 10% of the population.
Yep. Yeah.
Yeah, good.
Yeah. but we contribute a lot to the economy because all the minerals and ore and gold is in our state.
Oh, good.
Yeah. Now I'm just wondering, how do I share my screen? Do I have to keep my PowerPoint open?
Yeah, I think it would be better so people may follow.
Or do I just
there is a green box with arrow.
Yeah, that's
share screen. Click on it and choose your file.
Yeah,
you can.
I'll have to click on my presentation, right?
Yes.
Yeah. Okay. Okay, doke. So if you don't mind, I will just tidy that up and I'll temporarily leave.
And I will start in 1 minute.
Yeah, I'll just keep it open. Okay, you're going to start in 1 minute, so you don't want me to open it, right? No.
Yeah, be ready.
Okay, now how do I minimize this?
Every line, online education is challenging for people who are not used to.
No. I mean, we've done it. You know, it's just that it all depends on the system you use. My university has its own, so.
Now we have your photo.
I don't need that.
Let's start, Dr. Haider.
Yes.
Oh, where is my presentation gone?
Okay, let me introduce. Bismillah ar-Rahman ar-Rahim, and good afternoon, everybody. Welcome on board in this afternoon session, in which we will have 3 presentations. The first presentation will be age-related changes in energy expenditure and energy requirements, which will be by Dr. Mario Soares, and he's a physician, scientist with a formal grounding in human nutrition and physiology. He is an adjunct academic in Curtin University, Perth, Australia, with a cross appointment as an adjunct faculty professor, St. John's Research Institute, Bangalore, India. Dr. Mario has over 30 years experience in teaching and research in nutrition science across the globe. He has taught into the undergraduate nutrition and postgraduate dietetics degrees at Curtin University as part of the food and nutrition theme. As a nutritional physiologist, Dr. Mario has a long-standing interest in human energy metabolism and regulation of body weight from chronic energy deficiency to obesity. In particular, is the measurement of resting and postprandial metabolic events as they pertain to energy balance, insulin sensitivity, and endothelial function. Dr. Mario has served the Nutrition Society of Australia as a chairperson of its Perth chapter, and also was the director research training for the School of Public Health, Curtin University. Dr. Mario was co-editor-in-chief of the European Journal of Clinical Nutrition from 2016 to 2020, and has now assumed the position of editor-in-chief of the journal. Please, Dr. Mario, you have 20 minutes from now.
Thank you, Dr. Haider, for that nice introduction, and thank you for the organizers for this opportunity to share some of my thoughts on what I think is a very important and relevant topic going forward, as all populations in the world actually the percentage is increasing over 60 years. So I'm just going to try and share my talk for today, which is a slightly difficult one for. Is that now visible to everyone?
Yeah, it's clear. It's clear.
Yeah, okay, good. Thank you. So, what I'm going to do is basically give a quick overview of energy expenditure measurements and some of the pitfalls in the methodology, which those who are interested in the field should keep in mind, and then I'll tie them into to how to calculate energy requirements and what are the changes in body composition, etc., that takes place with aging. So I'll kind of flip-flop between the two. So to kick off, I mean, it's clear that to understand energy requirements, you need to have a handle on energy balance. And this is a very straightforward and simple slide, which tells you that, you know, if energy intake equals energy expenditure, then basically you're in energy balance, bearing in mind that measures of energy balance can be slightly tricky. Energy intake for most populations is the intake of carbohydrate, protein, and fat. And for those cultures which don't partake in alcohol, then it's even easier. And this has to be balanced out by EE, or total energy expenditure. Now we have three components. Resting energy expenditure, or RMR, is the largest component, and most of the talk will focus on this. Thermogenesis, usually diet-induced thermogenesis, is a small but significant 10%. And then the most variable component, obviously, is physical activity, which will range depending on the habitual activity of the client or population group under study, from 20% to 30% as an average for everyone. But obviously, people who are highly active, it can compose as much as 50%. So why do we study energy expenditure? I mean, there are a couple of factors. You may be interested in the physiology of risk of weight gain. You may be interested in how the body adapts to deficits or excesses of energy intake. Or, as is the purpose of this talk, you are interested in human energy requirements. Now, in this slide, clearly it's the same previous slide, just depicted a slightly different way. So you have an idea of the proportion of resting energy expenditure, which makes up a major chunk of total energy expenditure. 10% is diet-induced thermogenesis, and then you have physical activity. So clearly, if you're interested in energy requirements and you're going to measure energy expenditure, then if you have a very good handle on resting energy expenditure and physical activity, then most of your job is done, because you will have captured about 90%. There are several factors. I'm not going to go into the details, but suffice to say that the ones marked in red, weight, height, age, gender, are socio-demographic and anthropometric indices, which are generally used in the literature to actually predict REE, because some labs don't have the required equipment to do so. And it's a common trend that predicted REE is used for requirements, and there are issues with that. In fact, I would dissuade anyone from doing that till such time that we get equations which are very, very good. So our forefathers coined the term basal metabolic rate, which is, the least amount of energy and that is expended in man. In fact, you can almost say BMR is equivalent to the amount of energy you need to give someone to keep them alive. So from a kind of patient hospital-based point of view, once you got an idea of BMR, you at least know how many kilocalories they need to stay alive. But that is not good enough, because obviously you need to allow for additional energy for any activity, or if there's a disease process, or if there's fever, etc., etc. Now, in common in the literature is this kind of interchangeable use of terms, which I pinpoint here only because when you're actually doing these measurements, if your patient does not, or your client does not come to your lab or office to be measured and is resident there overnight, then what you will measure is the BMR, because it's measured soon upon waking. If they've had some physical activity, then obviously you're going to be measuring REE. And of course, if they actually, you make it so comfortable for them to be measured that they actually sleep during the measurements, you're actually measuring SMR, or sleeping metabolic rate. The reason I put it up here is you've got to be aware of what you're measuring and how your patient is reacting to the environment under measurement in the measurement period, because you can get a range of up to 10% in just the measurement. Okay, so indirect calorimetry is the usual process for the measurement of energy expenditure, and these are the usual setup where we conduct the respiratory gases, either from a nose clip and a mouthpiece or a face mask or a canopy, into a machine which has both the analyzers for oxygen and carbon dioxide. And then from that, you can deduce rates of energy expenditure. The reason I put it up is depending on what equation you're using to predict and what method they have used, the deviation between what you obtain and what you predict could well depend on the methods involved. Suffice, at least from my point of view, if you actually want to measure a good RMR or REE, it's best to use the canopy system because it does not encumber the patient as much as these two use, though if you give your patient sufficient trials or training, you can get them to breathe quite easily without any hyperventilation using a mouthpiece and face mask. Another aspect I'll just briefly touch upon before we actually go into the details is just, it's always a good habit to question your patient about their weight status prior to coming to your measurement, because we now know that even a small positive energy balance or a negative energy balance actually will change the measured RMR in a direction far greater than what is expected. It increases with overfeeding and decreases with underfeeding. Okay. Just may try and move this to one side because it's coming in my view. Okay, so I hope the slides are clear to everyone because this icon is disturbing me. Diet-induced, yeah. So diet-induced thermogenesis basically is nothing more than thermogenesis is heat production due to a diet. Very few people actually measure diet-induced thermogenesis because it means monitoring the person for all the meals of the day. Most people actually look at either nutrient, that is single nutrient induced thermogenesis, or meal induced, where you give them a fixed mixed meal of mixed macronutrient proportion of known kilocal, and then you monitor it over time. And there is a obligatory loss of energy in assimilating that meal. Hence, every meal or every mixed meal that you give a patient, there'll be, depending on the composition, some amount will be lost because that's what the system demands to actually process that meal and absorb it. And there is an adaptive component to it as well, which can be switched on depending on if in times of overfeeding or in times of underfeeding, the sympathetic nervous system can upregulate or downregulate metabolism so as the person can come into energy balance. So this is a very simple scheme. We measure the increase in oxygen consumption above the baseline. You want to capture this area under the curve. And depending on how many kilocalories you give the person, this time of observation could be 2 hours for single nutrients, or could run into several hours if you've got a high fat meal. The key point to remember is always try and capture the entire thing. There are too many papers in the literature which actually take a shortcut and then report 75 or 80% of the area under the curve, so as to speak. And because this is a very small component to total energy expenditure, the errors kind of creep in if you're going to then just do 80% of the meal. Physical activity is a very tricky component, and I would encourage anyone interested to begin to measure physical activity in a more systematic way, because one, it's highly variable, as I said to you before. But if you look at the available methods available, you can quite clearly see that the precision of the technique actually depends on the ease of assessment. The easier the measurement, like self-reports, or movement sensors or heart rate, then the precision tends to be less. If you want accurate measures, then in my book, the best one so far is the doubly labeled water method, which I know is rather expensive and restricted to a few labs, but it provides a very good idea of someone's habitual physical activity. Now, healthy aging, a lot has been written about. We can expect, large numbers of people in all countries of the globe who will live well beyond 60 plus. And the question is, is increased life expectancy increased health? And the answer, short answer is no, because we have so many chronic disease which kind of creep in as people get older, and also their roots may be prior to reaching the so-called, elderly cutoff. They tend to manifest more in the older age groups, be it CVD type 2. And so it becomes, in my book, very important to have an accurate estimate of how much food do people require, especially the aging population, before we go on to actually talk about, what is the macronutrient composition of that meal. So the basic principle of energy requirements, if not clear by now, is very simply, if you're in the process or have the option to measure total energy expenditure, then obviously you will do so by using doubly labeled water. If you don't, then you can actually measure resting energy expenditure and have a good assessment of physical activity and put the two together, making a kind of 10% allocation for thermogenesis due to diet, and then you will arrive at an estimate. The key thing is measured is always better than estimated, because, and I will show you in the next couple of slides, that estimated is never even, at the best you can expect plus minus 10% of the true value. And even that is only going to be in a small percentage of the population under study. So we've not done too well really in predicting resting energy expenditure or total energy expenditure. Okay, so this summary slide basically says, if you don't have access to doubly labeled water and you can't measure total energy expenditure, 24-hour room calorimetry is possible. It requires specialized labs again, so the same problem as doubly labeled water. And the other disadvantage may be that when the patient is or volunteers residing in that room, it may constrain them in terms of their physical activity relative to what they may do in a free living state. So we're actually left with either measure resting energy expenditure and multiply it by the physical activity level, or what is commonly done to predict resting energy expenditure and multiply it by the physical activity level. Now, there's a whole range of activities in the published literature where you can get to know what is the physical activity level for a group of people who are engaged in a variety of activities. And as you can see from this slide, 1.2 allows you barely any kind of activity, right down to highly active, which is roughly around 2 to 2.5 times their requirement. And by now you would have understood that resting energy expenditure actually equals 1. So if you have resting energy expenditure and you multiply it by, say, for a person who's engaged in heavy physical work, it could be resting energy expenditure multiplied by 2 or 2.5, or if you're an elite athlete in the Tour de France, you may even require 3 times your resting metabolic rate as your energy requirement. So this is a very nice paper, a systematic review done by Porter et al., where they actually collated individual data. Most systematic reviews tend to look at group means. They actually contacted the authors and got the individual data of doubly labeled water data in older adults. And they tested more than 17 prediction equations. They used a fixed physical activity level for men and women, given here. And what is shown here is only the 3 best equations. And if you look at the mean bias, bias is the measured value minus the estimated or the predicted value, then the bias is actually very, very good. But if you actually look at the limits of agreement, they are huge. And anyone who works in clinical nutrition in a hospital-based setting knows immediately that, if you're under-predicting or over-predicting by, 2,700 kilojoules per day, your patient is going to be in trouble, right? And so the same applies to free living population groups, because that's DLW data on free living populations. And so we have not done too well. The mean may be very good for the group, but individual people will be worse off. More importantly, if you have a mean bias You know, sometimes it's possible to apply a correction factor, but even that is not possible, because if I just look at the Mifflin data, which was the best equation so far, that's the mean for the whole group of only 24 kilojoules, which is nothing really in terms of, you know, total energy expenditure. There is a linear relationship between measured total energy expenditure and the bias, suggesting that those at the lower end are underestimated, those at the upper end are overestimated. And so this proportional bias, whether the person is 65 to 80 or greater than 80, it's across the board. And so you can't really apply a correction factor, even if you had this very good equation, which seems to fit these individual data, okay? So what are we left with? We're left with measurements of REE wherever possible, but global equations are there. And, you know, without going into the details, the global equations, which were first commissioned by the FAO, WHO, and the United Nations University in '85, they're commonly called the SSJ equations. They were flawed because they had a database which was a particular Italian database, which had very high physically active men, so it didn't resemble normal populations. And Jaya Henry then came along and, you know, deleted that data set and added more data, you know, 20 years later. And so I think as a global equation, this may be better. But remember, all these equations actually just use age, gender, weight, height, and, you know, depending on the categorization by the author, you will get age groups and gender groups to apply. They're based on weight, and weight is not a good, it will relate to energy expenditure, but it's not the best. And so we've not done too well in either
Mario, 2 minutes.
Yeah, okay. So what do we do? You know, we know that resting energy expenditure, which is the greatest component, declines with age. The greatest decrease with age occurs in the fat-free mass component. And if I can just show you that, it's not only the fat-free mass component, but it's also the composition of the fat-free mass component. Now, this slide is an old one. This is just to tell you that, you know, if you're basing it on weight, and I'm using BMI as a kind of surrogate for weight, you can have two people of the same BMI, but totally different percent body fat if you base it on DEXA. And you can do the same thing. You know, you can even look at fat-free mass, and if you actually split fat-free mass, then the highly active organ tissues, which only contribute a small percentage to body weight, actually make up a very, very large contribution to energy expenditure. And skeletal muscle mass at rest, which is, you know, a major portion of body weight, actually contributes very little. Remember, it's at rest, okay, not during exercise. And so clearly you need to, I'm going to skip because I've got a few minutes left. You clearly need not only to measure body composition and fat mass and fat-free mass, you actually need to partition out that fat-free mass into skeletal muscle mass as well as organ tissue mass, if you actually want to get a good prediction handle on resting energy expenditure. Okay, and so this is just data, again, data from Giesler working in Manfred Muller's lab, which shows that even if you have the best techniques, the problems that arise from the best techniques is sometimes we don't know what is the unit coefficient for organ tissue mass, because even that changes as someone ages. And so, you know, there's a lot of work to actually to be done. So if I was to summarize what happens with aging, then this is the best slide. It shows quite clearly that with aging, there's a decrease in total energy expenditure, both in men and women, more in men than in women. The greatest decrease in men is due to a decrease in RMR and in physical activity, whereas the TEF, or the thermogenesis component, is roughly the same. there's lots of, I mean, you know, at the end of the day, even if you had all of this, you would not explain all of RMR. There'll be about 10 to 12% of RMR left unexplained. And that is where factors like, you know, insulin resistance, HOMA-IR, CRP, thyroid stimulating hormone, and from our lab, even vitamin D, can all make a contribution to predicting RMR. And therefore, the way forward is actually to include those variables. Okay?
Dr. Mari, time is over.
Okay, so in conclusion, sorry for this, people. In conclusion, we need to have more data on good measures of resting energy expenditure, total energy expenditure, and body composition. And if you actually look at the worldwide database, then in my book, these are the countries who are not represented in the WHO database. So I would encourage workers in these regions to actually set up labs where you can measure resting energy expenditure or total energy expenditure and body composition. Thank you for your time.
Thank you so much, Dr. Mari, for the interesting presentation. I think we will keep the questions at the end of the session, and we move to the second presentation, which is by Prof. Ahmed Al-Khatib. He's going to talk about age-reversing bio nutraceuticals and exercise approaches from molecule to lifestyle. Prof. Al-Khatib, he's a world-leading expert in the health and nutrition and exercise science. He's a full professor at the University of Taipei in Taiwan and side in UK. His achievements included successful leadership and development of a major research and academic programs at several UK and international institutions. He's headed the epidemiology and public health at the Dasman Diabetes Institute in Kuwait, and part of the Non-Communicable Diseases Global Consortium on cohort studies. His research focuses on preventive and treatment role of nutraceuticals and exercise on diabetes and associated chronic diseases, and has developed a number of novel lifestyle prevention approaches. He also developed age-related treatments using functional foods and physical activity models relevant for global regions, including cohort from the GCC, England, and Scotland, and East Asia. Prof. Al-Khatib, he's a fellow of the Royal Society of Medicine and executive of the Asian Nutrition Society for Sports and Health, and fellow of the UK Higher Education Academy. Dr. Khatib, he has over 100 research publications. Please, Dr. Khatib, we will have you for the coming 20 minutes.
Thank you very much for the kind introduction, Dr. Haider, and I'm just going to share my presentation. I hope you can see that. I hope that is clear. Okay. Is the screen shown? Okay.
Yeah, it's clear.
Okay, thank you for the kind introduction. I'll be talking about age in terms of prevention. So how could we reverse and prevent the aging process and the chronic diseases particularly associated with aging using nutrition and exercise, and in particular try to segment some of the nutrients and the derived nutraceuticals in terms of their preventative benefits and how they're linked with aging. So in terms of the prevention, we all know that there are many different approaches to prevent disease, particularly the age-related diseases. We know the main mortality causers in terms of the in advanced age are the cardiovascular disease, cancer, diabetes, hypertension, and then some pulmonary diseases. So in terms of the prevention approach, it's important to take an approach that targets high-risk individuals, and that approach allows us to have a more personalized approach and is highly effective. However, other approaches might be relevant in the early phase, where we develop an infrastructure on a population-based. Certainly from my experience in various countries, in Kuwait, taking a population-based diabetes prevention program that targets large sectors of the population is effective in the early phase, because it targets those with risk factors, those with obesity, which represent quite a large section of the society at all ages. However, in terms of being able to go deeper in terms of understanding the biological mechanisms and understanding how we can personalize any treatment or any behavior, it's important to target those who might be at high risk from the population, including those within pre-diabetes or with advanced obesity, or the specific older groups. And I'll give you some examples later on. I put some of those in a recent review. I've been invited by the European Medical Journal to really describe how we can personalize the diabetes lifestyle prevention in terms of both the exercise and physical activity and in terms of nutrition. And there are quite a few models, including the one I presented here. So let's talk about diabetes, because it's prevalent in the GCC region. And my experience, certainly in the region, have shown that it's important to really apply the cause in terms of the prevention. So the cause of diabetes is a lifestyle disease, essentially, particularly type 2 diabetes. Over 90% of the cases are type 2 diabetes. So using lifestyle, which is the cause, to prevent diabetes has been shown for many years, and it's come to the forefront through the major prevention programs in the US, in Finland, which is one of the most or the earliest evidence that have shown that you can prevent diabetes by applying lifestyle. I've been fortunate to have worked with the leader of the Finnish diabetes study as well when I was in Kuwait, Professor Tomilito. So all of those interventions at different countries, they have shown a consistent prevention up to 71%. So the consistent results have shown in two of the largest prevention programs, 58% across all populations, and up to 71% in the older and the aging groups. So that is clearly a clear evidence to show that it's more effective as we advance with the age. The interesting American study, the US Diabetes Prevention Program, have compared the diabetes medication metformin compared with lifestyle, which consisted of physical activity and nutritional interventions, basically in the form of modifying those behaviors. And they've shown 58% reduction in the diabetes risk of the diabetes incidence, which is quite striking, having seen the same benefit earlier in the Finnish diabetes study as well. So clearly shows that lifestyle is actually even better than medication, or at least similar in terms of the prevention effects. And this is done in quite a large number of individuals that have been followed over 5 years initially, and most recently in the recent publications over 15 years. So we know that there is evidence there about nutrition and exercise. But if we segment slowly from the prevention and go slightly deeper into what sort of dietary approaches, and later on I'll show you what sort of nutraceuticals that can be derived from these healthy diets. So the Mediterranean diet, we've all heard of it, and we've heard of the PeriMed study, or the PREDIMED study, which is the large study has been conducted in Spain and looked into the benefits of the Mediterranean diet in both preventing cardiovascular disease and in terms of the preventing mortality and cardiovascular mortality. So they followed large groups of people over 5 years and provided them with various approaches. One of them is reducing or caloric restriction, which is the control group. And then the other two groups, they've consumed the Mediterranean diet, either with olive oil or with nuts, supplemented, mainly tree nuts like almonds and other types of tree nuts. And they've shown that the Mediterranean diet groups, the two groups that we can see here on the top figure in the bottom, they have introduced after 5 years, after 4 years, they've shown a significant reduction in the both the cardiovascular incidence rate and also a reduction in the mortality rate. But more interestingly, that I take from this, if we look at the mortality, total mortality or cardiovascular related mortality, we can see that those with olive oil benefited a little bit better, or they've had less risk after the 5 years compared with the two other approaches, when the control and the with nuts. So we can see that what might work for reducing specific risk endpoints in terms of mortality might be slightly different. So we need to take that into consideration. So we look at the age-related prevention and also the disease-related and the age-related diseases, which might work slightly differently. So here, in terms of the evidence about the Mediterranean diet that has already been established, we were the first, or actually one of the first groups, and certainly in the UK, we did the first work in terms of combining Mediterranean diet almost 10 years ago, combining the Mediterranean diet with exercise to reverse the age-dependent vascular activity. And we've looked into various biomarkers, lipid profile, particularly focused here on the endothelial function and vascular, which indicates the vascular activity at the microcircular and macrocircular levels. And we've shown that the combination of exercise with the Mediterranean diet, adoption of Mediterranean diet, intervention using those two approaches would certainly elicit significant benefit in terms of the endothelial function, enhancing the microvascular activity, which reflected in various better outcomes overall that are reflected in the cardiorespiratory function of the body and the metabolic function, of course. And we've seen earlier the example about enhanced energy expenditure and enhanced resting metabolic rate. In terms of the risk groups that we've looked into, we've also looked at those who are in the older age, those who are in the post-menopause state, and we've shown consistent finding that the combination is actually slightly better. However, either exercise or exercise in the Mediterranean diet makes a significant risk reduction in terms of the age-related or post-menopausal-related risks in this other study. Now, what components are beneficial of the Mediterranean diets? So yes, it's all good applying a holistic approach, which I definitely promote. However, it's important to understand the benefit of each component. So the Mediterranean diet is characterized by large intake of fruits, vegetables, olive oil, for example, legumes, nuts, moderate intake of fish and dairy products, and low intake of meat and red meat. So it's certainly what it appears that it certainly is quite high in carbs. However, the healthy type of carbs and high in fiber. At the same time, the specific components, they have significant benefits individually, and collectively they introduce those risk reduction benefits in terms of various diseases. So ideally, if we're able to have a nutraceutical derived from all of these components would be ideal. However, that might not be possible in the present. So in the meantime, we segmented the benefits of specific nutrients within the Mediterranean diet. So the first, we've looked into what benefits are there in the olive oil and how could that, combined with exercise or physical activity, augment or increase those benefits. And we presented the first review to actually make interesting observations from previously published studies about that we can actually augment various benefits when we combine olive oil intake with an active physically active lifestyle, particularly anti-inflammation mechanisms, antioxidation, and some interesting anabolic benefits that enhance the muscle atrophy. So the olive oil contains, particularly extra virgin olive oil, contains polyphenols or biophenols, which are very important, particularly hydroxytyrosol, tyrosol, and various other alpha linoleic acids and various other nutraceuticals that including also the monounsaturated fatty acids, which are implicated in various mechanisms, physiological and biometabolic mechanisms that give us these healthy benefits and lead to these healthy outcomes. Of course, there are other functional foods in addition to the Mediterranean diet, could be also related to prevention of specific diseases. We've summarized the evidence of diabetes prevention in terms of various functional food components, including specific herbs, beverages, teas, and including main components of the Mediterranean diet as well in this process. And the most important thing that I've always promoted is combining those with exercise, with physical activity, in order to get added benefit in terms of preventing the disease. So if we look at the micronutrients as well from certain foods, certainly the diet is the best source of taking the macronutrients, and less so of in terms of supplementing those, unless the risk is increased. So in this table, which is part of the European Medical Journal paper in 2020, we summarized some guidelines, some recommendations based on exercise and also based on nutrients. And in this table, I've shown the macronutrients recommendations, and those have been shown in the first in the functions and the effectiveness in terms of physiological function, and secondly in terms of the recommendations for supplementation. So supplementation may be effective for some minerals and vitamins like magnesium in advanced cases, particularly in ameliorating insulin sensitivity, reducing the risk. In vitamin D sometimes. However, dietary intake is always recommended first before resorting to supplementation. Unless the studies suggest that unless there is a specific risk in the form of a disease trauma, i.e. in advanced situations like advanced diabetes with complications, then certain supplementations would be effective, particularly D, C, and magnesium. So for those who are wondering what is functional food and what are the in comparison to nutraceuticals, any food could be considered functional when we take it.
Khatib, you have 4 minutes.
Yeah, but the nutraceuticals are the chemicals or the biochemicals that can be ingredients that can be extracted from those foods. So in the form of polyphenols, in the form of monounsaturated fatty acids, and available in various components, and also those derived from the herbs. So I move now to talk about specific longevity or age-related mechanisms and how certain nutraceuticals enhance those. So in this study, we've shown in Taiwan, we've shown that the supplementation with ginseng component RG1 is able to replenish depleted satellite cells, which are important for the aging process. Those are stem cells that replenish the aged cells within our cells. And we looked here, we can see here on the left the muscle fibers and how those who didn't have the supplementation have higher number of those aged cells that weren't removed, were only removed when supplemented with RG1. And that is an important, oxidative stress is an important mechanism for aging. And certainly being able to play in those mechanisms and try to reverse those is an important factor for longevity. Of course, further work is still needed, and sometimes certain nutraceuticals might be beneficial for certain diseases, like lowering the tumor size in this study that we've shown here, but not necessarily making any difference in terms of longevity. So they live the same lifespan, those animals that we've looked at, from 6 to 24 months. But those who consumed the supplement that we gave here, which is derived from deep ocean minerals, were able to have to develop less tumors. Other supplements that are implicated in the oxidative stress, which explains one of the theories explaining aging, is the anserin, which is a dipeptide available in the fish, like salmon, available in chicken breast, available in various other protein type rich foods. We've shown that it makes a significant effect on specific antioxidation, increasing the antioxidation, looking at SOD and various other markers, and explaining interesting physiological mechanisms. There are also, of course, other herbs that we here often consume: green tea, yerba mate, for those who heard of it. It's South American, also popular in the East Mediterranean region, or in the region where you are. And it has interesting benefits both for enhancing metabolism, particularly fat metabolism, particularly when it's combined with exercise. So in this, we've shown, the colleagues in Taiwan have shown, the combination with exercise induces better obesity reduction benefits in terms of fat metabolism and weight loss as well, in independent studies. We've shown here using techniques of indirect calorimetry at different exercise intensities that yerba mate increases fat metabolism and also enables better removal of blood lactate concentration. And these are some of the mechanisms how yerba mate could be beneficial for obesity, how it could be beneficial in combination with other behavioral elements like mood state, like satiety, with or without exercise, and how it can be useful when we combine it with other ingredients. About 30 different ingredients we've looked at here, and we've shown that, yes, it's conducive to being combined in terms of give us this fat loss benefits, increasing substrate
Time's over,
utilization.
Dr.
Yeah, just to wrap up here. And here we looked at the caffeine used to ameliorate the obesity endotoxins, which is quite important mechanism for disease prevention. Other nutraceuticals can be implicated in coronavirus prevention, and we've summarized some interesting evidence about antiviral functional foods segmented in our recent paper. And I put all of those in this model, which I call the exercise and nutrition lifestyle prevention model, where we take different approaches and different techniques to reach our secondary outcomes and primary disease prevention outcomes. So that's the end of my presentation, and thank you all for listening.
Thank you so much, Prof. Khatib, for the valuable presentation. And to move to the next presentation, our guest is Prof. Louise Dye. She will talk about obesity and aging brain. What are the risks? Prof. Dye, she is professor of nutrition and behavior, and leads the nutrition and behavior group in the Human Appetite Research Unit in the School of Psychology, University of Leeds. And she's academic lead for the University of Leeds. And she is interested in the research of functional foods for well-being, stress management, mental health, and cognitive performance decline in altered metabolic stress states, for example, obesity, type 2 diabetes, and genetic disorders. Please, Prof. Luis.
Good afternoon, everyone. I hope you can all hear me. I'm Louise Dye, and I've been asked to talk at this training event on obesity and the aging brain. So I'll give a bit of credit to my team, Neil Boyle, Claire Lawton, and Katie Adolphus, who've done a lot of this work with me, and we're all at the University of Leeds in the Nutrition and Behaviour Group. So what I was going to talk about this morning, or this afternoon, sorry, is the impact of obesity on cognitive function. You'll have the opportunity to ask questions during this talk if you wish, and you can paste them in the chat box as we go through. Okay, so without further ado, I'm going to talk today about obesity in midlife and how that might relate to dementias such as Alzheimer's disease, and then to consider the kind of consequences of obesity in midlife, like impaired glucose tolerance and type 2 diabetes, and how those impact on cognitive function, and also how nutrition could have a role in this. So can we actually improve obesity and diabetes related cognitive impairments by weight loss or nutritional intervention? And I'll touch, because I think it's quite pertinent, on the role of stress in obesity and cognitive function and cognitive decline. So just go forward. So I think what this slide is a little bit old now. It shows us is the proportion of men in the upper quadrant and women in the lower half who were overweight or obese in 2008 or the nearest year. So it's quite old data now, but what you can see is startling proportions of adiposity across Europe. And I think if you look at the fact that men are in position 3 and women in position 1, probably one of the only positives of Brexit is that the Europeans on average will get thinner when we leave. So the population's got fatter, it's also got older. And here you see the population curve, the demographics by age and sex in 2010 and the projections for 2060. So you see this middle bump here of baby boomers, 65, 69% of the population, who will live into older age, and then we'll have a very different curve. So what we're getting with that is an increased number of telegrams that the Queen has to write, but also potentially an increase in old age dependency. So what makes us dependent in old age is physical frailty, but it's also cognitive frailty. So we know, for example, that memory declines with age, but the decline is greater in some tasks than others. So things like riding a bicycle, which is a procedural memory, knowing how to do something, doesn't really change with age. You remember how to drive a car, how to ride a bicycle. Your general knowledge doesn't change. You might forget some things, or you might be a bit out of date in terms of laying down new memories. So when you do ask people to do a cognitive test that asks them who is the Prime Minister, they might remember a previous one and not the current one if they have some kinds of dementia. But what really does change is episodic memories. So problems with episodic memories are most common in mild cognitive impairment and in early Alzheimer's disease. And what's also common for lots of us as we age is a decline in prospective memory. So remembering to do something is quite common. In people with Alzheimer's disease, that can be things like remembering to eat. So mild cognitive impairment then is distinct from normal general cognitive performance. It doesn't involve dementia according to clinical criteria, but it does interfere a little bit with normal activities of daily living. And people show a subjective memory complaint. They say their memory is not as good as it used to be. However, they don't show objective. They start to show, with mild cognitive impairment, you start to show objective memory or other cognitive domain impairments. And what you see here on these PET scans is the progression from normal cognition through mild cognitive impairment through to Alzheimer's disease with less and less activation in the brain. So we know that as we age, the prevalence of dementias increases, and the Alzheimer-related dementias also increase. So by the age of 80, over half the population is likely to have some form of dementia. Vascular dementia then is about 17% of the dementias, and that affects blood vessels to parts of the brain that are involved in memory, language, and thinking. And this type of dementia results from damage or blockage in these blood vessels, which effectively decreases blood flow to the brain. And the relative risk of vascular dementia in those people with type 2 diabetes is 2.49. For Alzheimer's, the relative risk is 1.46. And Alzheimer's disease is a progressive problem. The earliest changes might even occur 20 years before diagnosis. And age-related changes in glucose regulation have been proposed to be a risk factor for Alzheimer's disease. And when we look at patients with Alzheimer's disease, around 80% have problems with glycemic control. And that's led to some people proposing that Alzheimer's disease is a kind of type 3 diabetes because it's a metabolic disease, and what we see is impairment in brain insulin responsiveness, in glucose utilization, and in energy metabolism, and also changes in insulin-like growth factor. So the mild to moderate stage of Alzheimer's can last from 2 to 10 years, and the severe stage from 1 to 5 years. And losing your cognitive functions makes you much more dependent in your older age. So old age dependency is defined as the share of people aged 65 and older compared to the population in working age. There's a difference, though, between being dependent. So old age dependency like this is a population type measure, and you can see various parts of Europe with increasing levels of older age dependency. As we get an aging population, we'll have more people retired relative to people working, and that's fine if they all stay fit and healthy. But as soon as they become physically or cognitively dependent or impaired, then that causes all kinds of social, economic, and health burdens for the whole population. So what's obesity got to do with this then? So there have been quite a number of studies that have compared obese adults with normal weight adults. This is some stuff from John Gunstad's group in the US, and they find that obese adults perform worse than normal weight controls on measures of learning and memory and on frontal lobe tasks. So learning and memory are fairly obvious. Frontal lobe tasks are things like executive function, so planning, making strategies, complex attention, and psychomotor speed. And there's been some argument that impairments in this kind of decision-making capability might also contribute to poorer dietary choices. So a recent systematic review looked at studies looking at the relationship between obesity and cognitive function, and in the 17 articles that they found that they included, they showed that impairments were evident in all cognitive domains, but the quality of the studies was so poor that, and the methodological limitations so great, that they had to conclude that there was insufficient evidence of a reliable and valid association between obesity and cognitive function in midlife. However, Marioni et al. In 2016 studied Generation Scotland, which is an ethnically homogeneous cohort study in Scotland. And they did genome-wide association studies, calculating polygenic scores for traits like BMI and cognitive function. And while they found that the correlation between obesity and poor cognitive function was quite small. What it showed was a high BMI was associated with lower cognition. The BMI-specific genetic variants accounted for 42% of the variance in cognitive function, but cognitive-specific variants only accounted for 0.08% of the variance in BMI. And they also found that 7 common genetic variants were associated with both traits, which is more than you'd expect by chance. So what that tells us is that there are shared genetic contributions to BMI and cognitive function. So does it matter where you're obese? So does it matter what your body composition is and where you store your excess fat? So it's really the question about whether you're an apple or a pear. And we talk about that in terms of waist-hip ratio. So central obesity is reflected by a waist-hip ratio greater than 0.85 in women and 0.95 in men. And that reflects the presence of central fat in the abdomen. That can occur independently or as a symptom of metabolic syndrome. And stress can actually feed into this relationship. So under stressful conditions, you release glucocorticoids. Glucocorticoid excess leads to abdominal fat deposition by blocking the action of insulin, and that promotes further fat deposition, which increases your risk of impaired glucose tolerance and type 2 diabetes. So repeated exposure to stress exacerbates that vicious circle. So we've looked, there've been some studies then that have also looked at waist-hip ratio and hippocampal volume. So most of your memory processes occur in the hippocampus in the brain. And here's a study that showed that waist-hip ratio was inversely associated with hippocampal volume, even when you adjust for age. So the higher the waist-hip ratio, the smaller the hippocampal volume. And then we've looked at stress exposure in our laboratory. And as I said, we've got this vicious circle where central obesity co-occurs with glucocorticoid excess, and we think that can lead to elevated responses to stress. So in this study, we looked at 66 men and women who were high or low waist-hip ratio. They were middle-aged adults, around 46 years, and we gave them a series of cognitive tests. And what we hypothesized was that those with a high waist-hip ratio would exhibit greater cortisol response to stress and show poorer cognitive performance. Exposure to stress and increasing waist-hip ratio were specifically associated with poorer performance on memory tasks. In this case, the one shown here is spatial recognition memory, but also on paired associate learning. An increasing waist-hip ratio was associated with improved learning when they weren't under stress, but poorer performance in those that were exposed to stress. So here what you see is that an increase in waist-hip ratio was associated with a reduction in performance in spatial recognition for males under stress, but there was no effect when they had no stress. So Selborn and Gunstadt have proposed a number of obesity-related neuropsychological changes. And these are things like those factors associated with obesity, like hypertension, diabetes, and sleep apnea. And then the pathophysiological factors that underlie those, which are compounded by age. And they propose that this leads to changes in the brain in terms of frontal and temporal lobe atrophy, white matter, and metabolic and blood flow alterations. And that those lead to cognitive dysfunctions of the type I've just shown you, but also to behavioural outcomes like decreased initiation, poor self-monitoring, inhibition, adherence is impaired, and also mood disturbances, and that those things taken together can elevate your risk of cognitive decline in terms of vascular dementia and Alzheimer's disease. So let's take a look at diabetes then. In the UK, there are around 4 million people diagnosed with diabetes and around 1 million who are undiagnosed and therefore unaware. And in 2018, it was almost 250,000 people that were diagnosed. We estimate those rates will go up to 5 million by 2025, and 90% are type 2 diabetics. And we're seeing an earlier onset of type 2 diabetes. There are some genetic predispositions, so South Asians and Afro-Caribbeans may be more likely to develop type 2 diabetes, and that's one of the reasons that we see a very high prevalence in Bradford, where something like 1 in 10 of the population has type 2 diabetes. So in terms of cognition then, type 2 diabetes is associated with cognitive impairments, and those impairments reflect a kind of accelerated rate of cognitive aging. And the same reduced hippocampal volume that I showed you before in relation to wasted ratio is also seen in type 2 diabetics. But it's very difficult to tease that apart from normal aging. So if we look at middle-aged adults, we see modest decrements in specific cognitive functions. And in older adults, those decrements are much more widespread. They impact more cognitive functions, things like processing speed, attention, and executive function. And again, this is a study that we did in our lab where we looked at type 2 diabetics, and we compared them to normo-glucose tolerant adults, matching them for education, age, depression, and IQ, which are all things that should influence cognitive functions. So ostensibly matched, differing only in their diabetes. And what we were able to see here was impairments in verbal memory, spatial memory, psychomotor skill, and executive function in the type 2 diabetics. And here's an example. This is a Corsi block tapping task. So this is a kind of spatial working memory where you see a series of blocks that light up on a computer screen, one after another, and you have to repeat the pattern. So it's a bit like the Simon game that kids used to have years ago. It's quite fast, and at each level, the number of blocks to remember increases. So it goes from 2 to 9. And what you see here is that the type 2 diabetes group made fewer correct responses for all levels except 2 or 3. So significantly fewer, significantly worse performance at relatively easy levels of the task. And that was part of a whole battery of tests. And I think what we have to worry about there is that impaired glucose tolerance precedes type 2 diabetes. And so it's likely that there are many, many more people with impaired glucose tolerance than are diagnosed with diabetes across the world. And here you can see the prevalence rates of impaired glucose tolerance in 2013, which is actually quite high. Excuse me. So what we know from epidemiological studies is that even with impaired glucose tolerance, so pre-diabetic state, there is a clear association with impaired cognition. But those are tests of association. So those tests of association then are not necessarily based on objective measures of cognitive function. So about 10 years ago, we did a systematic review to look at whether there were any studies which indicated whether there were impairments of cognitive function in people with impaired glucose tolerance using objective and sensitive measures of cognition. So these were studies in pre-diabetic states where people were losing their glucose regulation, but they were unaware and not on treatment. They were predominantly middle-aged samples, and there were few effects on cognition. And we thought, well, that's really strange. But actually, it's because the tests that were used in those studies were very insensitive. So quite often people use the mini mental state examination, which is a test for dementia. So you have to be really quite impaired to score poorly on that kind of test. And the kind of sensitive test that I've just shown you in the diabetics hadn't really been widely used. So we decided that we would do a study of impaired glucose tolerance. This is work that Dan Lamport from Reading, who's now at Reading, did as part of his PhD. And we recruited women who were aged over 35 to do ostensibly a breakfast study. But when they came in, we gave them an oral glucose tolerance test, and we were able to show that they separated very clearly into those with normal glucose tolerance, who you can see after 2 hours of an OGGT have returned to baseline, and impaired glucose tolerance, where they have a much elevated glucose response and they don't return to baseline. These people didn't know that they had impaired glucose tolerance, and we gave them a battery of cognitive tests. Now, we gave them 27 tests, so you'd expect 2 or 3 to be significant by chance. But actually what we could see was that the impaired glucose tolerance sample were impaired on 12 out of 27 cognitive tests, and they were particularly poorly poor performance on measures of verbal memory, in both immediate and delayed, and measures of spatial memory, visual spatial learning, as well as the Corsi tapping task that was impaired in the diabetic and psychomotor tasks. So it was very much along the same lines as the type 2 diabetes, not so severe, but the same cognitive domains were affected. And so what you've got here then are quite subtle impairments in prolonged concentration in what are effectively ostensibly healthy middle-aged women who had no idea that they had impaired glucose tolerance in the first place, so weren't doing anything about it. So that kind of brings us on to think about what can we do about it. And I think you will know as nutritionists that a healthy diet and exercise can actually halve the risk of developing diabetes in people with IGT. So a healthy diet and exercise should also be beneficial for cognitive function. So if we're thinking about cognition throughout life, what we're trying to do here is prevent cognitive aging and to prevent that accelerated aging that occurs with impaired glucose tolerance and type 2 diabetes. So it would appear then that midlife would be a critical point to intervene. So we know that midlife obesity is a risk factor for Alzheimer's disease and vascular dementia. We know it's significantly associated with cognitive dysfunction. And we know something about the mechanisms being related to the comorbidities of hypertension, type 2 diabetes, dyslipidemia, and so on, or maybe independent things like neural degeneration and impaired cerebral metabolism. So these are the possible mechanisms then. So reducing obesity could improve things like insulin and therefore glucose regulation, could reduce inflammation, reduce oxidative stress, and improve cardiovascular status. But there may also be direct effects of nutrients that could also improve cardiovascular status and improve neurogenesis or maintain neurogenesis. And I think it's worth looking briefly at those. So quite extreme then measure of method to reduce obesity is bariatric surgery. So in bariatric surgery, it's usually in the morbidly obese that this might be performed. And there's been a systematic review that has looked at 18 studies where cognition was measured following bariatric surgery. And what they showed was that cognitive function was measured by a number of different assessment tools, and it improved rapidly following the surgically induced weight loss in almost all of the studies. And the improvements were particularly in areas like memory, executive function, and cognitive control. So the kind of areas that we've already discussed as being impaired in impaired glucose tolerance and in type 2 diabetes. But interestingly, those improvements didn't occur in individuals who had a family history of Alzheimer's disease. So that might suggest something like an ApoE4 allele might mean that they don't respond. Excuse me. They don't respond as well to rapid dietary intervention or weight loss. So we can also look at whether obesity-related deficits in cognitive function could be reversed by less invasive methods of weight loss. And there are a number of studies that have shown that weight loss through diet and exercise can improve cognitive function. So one study of hypertensives on the DASH diet, so the dietary approaches to stop hypertension, combined with aerobic exercise and reduced calories, showed improvements in multiple measures of cognitive function. And the DASH is very specific. It requires counselling. It's quite an intense programme. And then other studies that show overweight and obese people who lost 14 kilos over 1 year showed improvements in working memory. So what that seems to show us then is that obesity-related cognitive dysfunction is partly reversible, but it's hard to say what the specific mechanisms were. Now, I really like this study. This study was done in octogenarians in Japan. And it was a 2-year intervention where they were given exercise, so things like Tai Chi, sort of gentle exercise, these are over 80-year-olds, 2 to 4 times a week, and instructions to increase their dietary fibre to over 30 grams a day. So that probably represents a doubling of fibre intake based on the average intakes in the UK population. And what you can see here is that the vast majority of the sample of these octogenarians were normal glucose tolerant anyway, there was a reasonable group who were impaired glucose tolerant and a smaller group of type 2 diabetics. I don't think you'd reflect those proportions if you sampled octogenarians in the UK. And so they looked at the impact of this 2-year intervention on fasting plasma glucose, on a 2-hour OGGT, on HOMA, measure of insulin resistance, on the mini mental state, a dementia scale, a cognitive test like the ones I've just shown you, and the block design test, which is exactly the one I just showed you. When you see a 0, you see no change. And there's no reason really why these normal glucose tolerant people would change. Here, a minus means that these things went down, which is a good thing because these are measures of glucose tolerance, glucoregulation, and that occurred both in the IGT and the type 2 diabetics. And we also see that in the type 2 diabetics, scores on the mini mental state and the dementia scale and the recall all went up. And in the IGT, that didn't change. But you wouldn't expect a dementia scale to change in people who didn't really have any evident dementia. And then the more sensitive measures improved in both. So what that's really showing is that dietary fibre and exercise improved cognitive function, and it probably did it via improved glucose regulation. I think people here will be familiar with the PREDIMED study. And the PREDIMED study took people who were middle-aged with high cardiovascular risk and looked at the impact of a Mediterranean diet with extra virgin olive oil or a Mediterranean diet supplemented with nuts versus their control diet. And although there have been some problems with the PREDIMED study, this measures of changes in cognitive function is really quite interesting, because what we can see is over the 4 years of the study, cognitive function in the control group declined on both memory, frontal cognition, and global cognition, and it was actually maintained. So there was no decline in either of the 2 active groups. So they all did better than the controls in terms of their cognitive functions. But the diet supplemented with nuts appeared to be beneficial for memory. The diet supplemented with olive oil appeared to be beneficial for frontal cognition. And really, this kind of averages them out, the global cognitive measure there. So another epidemiological study is the Nurses Health Study, which followed 16,000 women aged 70 plus for a long period of time, but measured their cognitions in 2-year intervals. And what they found was that women who had a high intake of anthocyanin-rich fruits and berries, so good source of polyphenols, anthocyanins, colourful fruits, when measured on cognitive tests, and it was the average of 6 cognitive tests, showed a slower rate of cognitive decline. So fruit and vegetable, particularly polyphenol-rich diet, was associated with better cognitive function. But the nurses who showed that better cognitive function tended to be more senior, which probably means they had greater education. They probably earned more, so they were more affluent, and they also engaged in other health lifestyle behaviours like physical activity. So it's very hard in those epidemiological studies to directly attribute that slower rate of cognitive decline to the polyphenol-rich intake. And then I mentioned at the beginning that one of the mechanisms might be that blood flow to the brain is associated with poorer cognitive outcomes. So here we see that cerebral blood flow is greater in younger people and declines with age, and that there's also a difference between people with preclinical Alzheimer's disease and controls in terms of their cerebral blood flow. And a very nice study has shown that supplementation with high flavanol, cocoa flavanols, can improve cerebral blood volumes. And then Ana Rodriguez-Mateas has also shown something very similar with blueberries. So ingesting blueberries, and there seems to be like an optimal dose here of around 500 or 600 milligrams, is associated with an improvement in flow mediated dilatation. And cocoa flavanols have also been shown to improve insulin resistance and lipid peroxidation. So those might be other methods where the vascular function of the brain can be improved by polyphenol ingestion. So The effects of nutrition on mood and cognition are actually quite subtle. So nutritional interventions with enduring effects can take a long time to manifest, which is why we rely very much on cohort studies or epidemiological studies. So in a study by Alan Danger, there was 6 months fish oil consumption, improved recall of word lists by about 1 word. And the authors concluded that that treatment had no effect. But if that's the one thing on your shopping list that you forget when you go to the supermarket, that's quite meaningful for you. And others have argued that a decreased recall of 1 word might be equal to 1 year of cognitive decline. So in terms of the micronutrients then, they're also important for cognitive function because they do things like address inflammation. So micronutrients play a role in inflammation, either through modulating cytokine production or by scavenging byproducts, so free radical scavenging, which reduces C-reactive protein. Antioxidant micronutrients also address and limit oxidative stress, and they could also directly influence neurotrophins. So polyphenols also modulate oxidative stress, but they also affect things like brain-derived neurotrophic factor. And we know that flavonoid fruit and vegetables increase BDNF in serum at the same time as improving cognition. And then the Suvimax study in France showed that 8 years of antidote, where they followed people for 8 years, showed that the people who consumed antioxidants showed improved verbal memory compared with placebo recipients over a 5-year period. So going back to the point I made before, that these things may take quite a long time to manifest. So what can we conclude from this? I think that it's very clear that type 2 diabetes is associated with poorer cognitive function, that there are domain-specific effects, and that we need sensitive cognitive tests, so not global function or IQ tests to detect these things, but really sensitive domain-specific tests. And that cognitive effects are evident, but they're much less severe in impaired glucose tolerance. I think obesity is associated with poorer cognitive function, and it's exacerbated by stress, particularly in those who deposit their abdominal fat around their waist region, so central obesity. And the causal factors that I think we've covered include glucoregulation, insulin resistance, and other factors. So it's really important to reduce obesity in midlife. That would be beneficial. And for all kinds of reasons, it's important to preserve cognitive capacity in aging. So where should we focus our research and our public health efforts? Well, I would say we look at the younger populations, and for me that now means middle age. So the 40 to 65-year-olds are probably the key demographic where we need to look at reducing obesity and maintaining cognitive function throughout life. So we definitely need to do longer prospective studies and know more about biomarkers, and also take into account potentially harmful dietary components. But it's important too to look at neuroimaging data alongside cognitive tests that are sensitive to diet. And that's not easy. You can't just take a test off a shelf and assume that that's going to be sensitive to a dietary change. It's also quite important to look at the additive or the synergistic effects of other behaviours. So physical activity could be a nice mediator, equally body weight change. Okay. And I think, it's also quite important to keep using your brain. One of the protective factors for Alzheimer's disease is speaking another language. Not all of us can do that, but we can do something about keeping active, eating a good diet, and everything in moderation rather than excess. That's the rest of my group. I presented something along these lines, a little bit similar if you want to read more about it, at a Nutrition Society meeting a few years ago, and there's a nice paper that's based on that. And I've also edited a very nice book called Nutrition for Brain Health and Cognitive Performance, which gives a good overview of lots of the things that I've touched on today. So I'd like to say thank you to the Nutrition Society for inviting me to run this training event, and I'm very happy to take any questions that anyone might have.
Thank you so much, Prof. Dai. And now we will open the floor for questions for Dr. Mario. Any of the audience has a question to the first speaker, Dr. Mario? You can put it in the question and answer. Okay, any question for Prof. Ahmed? I think there was one question, Prof. Ahmed Al-Khatib already answered it. Any further questions? Unfortunately, Prof. Dai, she has to leave us for another commitment, but if you can put the questions and you'll get the answer by email. The organizer will take care of the questions and will answer, send you the answer. Any question? Any question? Prof. Ahmed is ready to answer your questions if you have any.
I think the attendees, Dr. Haidar, got the informations of our speakers, and it's well received. We have received some comments that it was really interesting, informative, and wonderful session.
Yeah, really very interesting. And thank you so very much for the three speakers. And we got the maximum benefit from their talk. And we wish them all the best, and same to the audience. Thank you so much and have a nice time.
Before we end, Dr. Haider and Dr. Maria and Professor Ahmed, can we take a group photo with the three of you? Can you look at the camera? Dr. Haider, I think your background is quite a bit dark. Do you have a light on?
What about now?
Perfect. All right, Dr. Maria, Professor Ahmed, and Dr. Haider, can you please look at your camera on your laptop? At the count of 3, let's take a smile. All right, on 1, 2, 3, smile. Thank you so much, Dr. Maria.
Thank you. Thank you.
Thank you. Thank you.
Thank you.
Thank you all.
Thank you.
Lovely meeting you all. Thank you.
All right.
Take care.
Bye. Thank you.
Bye.
Good luck.
All right, attendees, please stay tuned. We have an upcoming session at exactly 4:30.
I'm trying to share my screen.
Just one moment, Professor Lily. When
it says, will this stop others screen sharing? Do you want to continue?
Yes, it was.
Is it yes or no?
Yes.
Okay, and I'm trying to find my slides here, and I just can't. It was on my desktop. Ooh. Gee, I can't find my slides. Okay. Now, is this okay? Can you see something?
No, not yet, Professor Lily.
Oh, okay. Tell me when you can. Kim,
Miss Andus, can you unmute yourself? Shall we start, Miss Andus?
Okay, as I was waiting for you.
Yes.
Okay. May I start now?
Can I start my video, please? Oh, yes. Okay. Thank you.
So which comes first, video or Kim?
It's okay, Miss Andus. You can start introducing Professor.
Okay, good afternoon, all of you. This is the last session, I suppose. Welcome to Professor Liliana Stojanovska. Professor Liliana is a professor at the Department of Nutrition and Health at the College of Health Sciences at UAE University. She's also emeritus professor at Victoria University in Australia and honorary professor at University in Thailand. Today she will talk to us about the women's health. An interesting topic, by the way, about managing menopause without medicine incorporating nutrition. So welcome to Professor Liliana.
Thank you very much indeed. Thank you, Ms. Harif.
I have to be short, I'm sorry.
Yes, thank you. Thank you to the organizing committee for inviting me to this symposium, especially Professor Leila and Professor Aisha, and everyone else, and Kim, and the whole team for making this possible. My presentation today is going to be on women's health and basically managing strategies on how to cope with menopause at the time of this reproductive transition via different modalities that we have. Basically, these are my objectives, and I will get started with the menopause issue. And the reason why I have spent many years doing menopausal studies is because half of the population of this on this earth are made of women. So we comprise 50 to 51% of the women of the world's total population. Hence, we have a whole subject or to study in gynecology and endocrinology, which is part of menopause. Menopause is defined as the last menstrual period, and it is a normal, natural process. It occurs at about the age of 50, 51, and it means loss of estrogen and progesterone. Well, we are all familiar with this slide where we've got cyclical production of monthly production of estrogen, as well as, I'm just trying to get my pointer. I hope I can find my pointer here. But if not, the basic production of estradiol and progesterone is cycling, and at the time of menopause, it is static. In other words, the hormones have declined, not to zero, but close to. In men, there is a steady state of estrogen to their life, but it is a very small production of estrogen. So what do we see at the time of our hormonal changes pre and post menopause? Throughout the reproductive cycle, we've got high levels of estrogen that begin to decline after the age of 40, and after the age of 55, 60, we've got a very low concentration of estrogen and progesterone. On the other side, we've got increased level of gonadotrophin hormones, follicular stimulating hormone, as well as luteinizing hormone. And this is the whole process whereby we see these disjointment of normal reproductive stage. So we go from normal reproductive stage to non-reproductive stage. So basically, the time of menopause has not changed throughout the centuries. It has remained anywhere between 45 and 55 years of age throughout different cultures and continents. What has changed is basically our life expectancy. We live now a lot longer than our predecessors. And the point of menopausal issues is because we live 25 to 35 years post-menopause. And these postmenopausal issues are really important to consider because that window of opportunity of 30 years post-menopause is very important for prevention of chronic disorders. So on a population level, 25 million women across the world currently experience menopause every year. And in the next few years, we'll have more than 1 billion women, which is 12% of the world population in postmenopause. So hence the importance of this topic of menopausal issues, symptoms, chronic and acute. About 60 to 80% of the women at the time of menopause and post will have mild to severe symptoms related to menopause. And about 20% of women will have only very small percentage of symptoms. So therefore they range from psychological, physiological, and also problems associated at the time of menopause, financial issues, family issues, etc. Menopause is basically at the time when we go into hormonal decline of estrogen and progesterone, is predominantly associated with hot flushes and night sweats. And just about every woman will experience to some extent these, but as I said, 60 to 80%, a lot more, and 20% very little. In addition to night sweats and hot flushes, women at the time of menopause will experience weight gain, muscle mass decrease, a lot of irritability and reproductive problems in terms of vaginal dryness, loss of libido. They might even experience psychological issues such as mood swings, irritability, depression, not sleeping properly, insomnia, wakefulness, etc. So plethora of symptoms that are associated with menopause. However, menopause is not only hot flushes and night sweats, it's a lot more to that. And it's the fact of the chronic diseases at the time post-menopause. Remember, we live 25 to 35 years post-menopausal year, and then we really need to make sure we are combating cardiovascular disease, diabetes, osteoporosis, depression, and Alzheimer's at the time of menopause. So it's important to prevent these issues. How do we prevent them? First of all, we've got therapies, and these therapies can be hormonal and can be self-managed therapies. The hormonal therapies are prescriptive, and then also we have a whole plethora of non-prescriptive, complementary and alternative medicine. While with the self-management strategies, we've got nutrition, and this conference is all about nutrition, exercise modalities, and lifestyle. When we look at the hormonal replacement therapy, we do know that they can be prescribed to women with severe symptoms for very short time, low dosage. And we do know that HRT basically decreases the prevalence of symptoms. It also improves bone fracture. But however, we know that the risks associated with long-term of HRT is basically increasing breast cancer and cardiovascular embolism. And these are different type of publications to support my conclusion. On the other side, complementary and alternative medicine are natural. They claim to be natural. However, the negative side is that they lack scientific rigor, and there is not much validation studies in terms of use. We are all aware that our pharmacies and some supermarket shelves are full of these complementary medicine and alternative medicine, from red clover to remiferin, and they sell very well because it's a multi-billion-dollar business. Complementary alternative therapy is basically more worth to these pharmaceutical companies than ordinary hormone replacement therapy, or shall I say prescribed. More than 50% of women have used one product or another of complementary therapies at the time of menopause. And the numbers have excruciatingly raised at the time when the WHO, which is Women's Health Initiative study, was published a few years ago, and looking that prolonged use of HRT causes breast cancer. However, most of the studies are short duration, and they're not really properly validated and not in large population. So what are these? Predominantly, most of these complementary therapies are type of phytoestrogens. So what are phytoestrogens? As we know, they're non-steroidal plant compound derivatives, and they have a very similar structure to estrogen. And that is when we try to see why they might be beneficial. The predominant component chemical is isoflavone, and when it is converted into gut, into diastein and equiol, then the actual structure of equiol to estradiol is very similar. Hence the reason that they really might have some beneficial effect. That is, they might act like natural estrogen. Where do we find isoflavors? Mainly in soy proteins, soy milk, soy flour, soybeans. And this is the concentration per 100 grams. But also we find it in fermented products such as kimchi, such as tofu, as well as miso. In addition to that, we find them in grains such as lentils, dry beans, chickpeas, alfalfa, and basically it is, as I said, in many, many fermented products. This fermentation occurs in the gut due to the bacterial acting on it. So what we did in our laboratory some few years ago, we did a very small pilot study. We looked at 36 menopausal women. We divided them into 3 groups, parallel study. We gave them 13 to 30 women, we gave fermented milk, soy milk. The others we divided into non-fermented soy and casein, which is a control group. We did that for 12 weeks. We measured several parameters, and this fermentation occurred with bifidobacteria. We also published this study in Climacteric. We measured several parameters, but what's important that we measure the hormones, and this is the baseline, and we found that with either the fermented soy or non-fermented soy, we have had a decrease from the baseline in these hormones. Remember, FSH and LH goes up at menopause, so this is decrease. This is non-significant decrease, a small pilot study, which again was published in Climacteric, and we concluded it's a small study. We need to increase the population number of people, and we also need to make it into a lot longer study. But nevertheless, it is still indicative that soy might be important. So how much soy do we eat? Not much. We take very little soy in comparison to people that live in Asia, to Asian women. There is a claim that in publications that Asian women have experienced a lot less menopausal symptoms, and most probably this is due to the fact that they consume soy throughout their life, not at the time of menopause, throughout their life. So therefore, their chemicals and chemical compounds in the gut is converted from diacetyl into equal, secreted into the urine, and the equal produces, which many non-Asian women are non-equal producers, so therefore it doesn't really have an effect on them.
Professor Liliana, just
yes?
a few minutes left over, few minutes.
How many?
Oh, just 5 minutes maximum.
Okay, so here are some of the other complementary therapies such as Promensil, but most of the studies are indicating that most of these complementary therapies are basically showing more than 60% of placebo effect. Black cosh is another one that is on the market, and we did another study with alternative medicine, maca, which grows in the Andes, and this is maca with aphrodisiac. And we showed that this particular study, although small, it improves sexual dysfunction in women when measured on a green climacterics postmenopausal scale. I want to base a few minutes on my nutrition because it's very, very important. Studies published in Lancet indicate that most of the cardiovascular diseases across the burden in 195 countries shows that people die as a result of these problems, cardiovascular type 2, due to a consumption of foods that are low in grains and high in sodium. Mediterranean diet is supposed to be one of the best for women, and basically due to the fact that it's got high fiber, low reduced intake of simple carbohydrates, and high concentration of vitamin D in terms of rich oil. It's important to understand that we lose weight, we lose bone mass, beg your pardon, at the time of menopause. So fight bone mass and muscle loss, it's really important. So we need to prescribe green vegetables, rich with calcium, rich with vitamin D, and to also have lean protein for loss of muscle. In addition to diet, does exercise work? Absolutely it does. Plethora of studies are indicating that it helps from everything, reducing weight, symptoms menopausal, improving cardiovascular parameters. What is important is that this particular study, done SWAN study of women health across the nation, they look at that over 7 years, and they studied that obese women, menopausal women, had impaired glucose tolerance as perceived progression of high risk in the metabolic rate. So physical activity is the only lifestyle factor that can protect us from high risk state. We showed yesterday at the presentation by Dr. Khaled that telomeres are shortened every time the chromosomes divide. So that's the reason for our aging and death and cancer, shortened telomeres. So exercise basically improves shortening of these, that is, combats the telomeres to shorten and therefore delays this process, and we can live up to 10 years more if we exercise. How much do we exercise? How much is enough? At least 5-6 times to moderate activity in various types of intensities. If it's more intensive, less time and duration, less intensive, longer. Apply 2-kilometer walk. Walk from the car to the supermarket. Do not park the car near the supermarket. My last slide, self-management, putting the brake on menopause. Happiness, in being involved in leisure, hobby, activity, any of these, from gardening to religion to dancing to meditation, all contributes to decreasing the stress, improving the longevity, and having us achieve the age of 90. Not one size fits all. Different women require different management strategies, but at this stage, women do live longer. They need to not only live longer, but healthier, and to be engaged in computers such as nanotechnology and other things that younger generation do. And with this, I thank you very much for your attention.
Thank you, Professor Liliana. That was an interesting topic, by the way. It seems bifidobacteria is getting more popular. I don't know if we have time to ask you questions. I could see only one question in the Q&A, but I think Prof. Liliana, she went through this answer, through the presentation. So I think if there's any question to Prof. Liliana, we could postpone to the last, session. As per now, I think I would move to the second presenter. We have actually my colleague, Dr. Hadi Radwan. Dr. Hadi Radwan, she earned her bachelor and master degree from the American University of Beirut and PhD from Teesside University. And she's currently an associate professor at the Department of Clinical Nutrition and at the College of Health Sciences at the University of Sharjah. Her areas of interest actually includes maternal and child nutrition. She was a PI of several research projects, grants. Today's presentation is going to talk about the pregnancy, pre-pregnancy body mass index and gestational weight gain. So welcome with me, Dr. Hadia. Her topics today, gestational weight gain and gestational diabetes among Emirati and Arab women in the United Arab Emirates, result from the MESC cohort study. Welcome, Dr. Hadia.
Thank you so much, Ms. Sundus, for the nice introduction. I would like to thank the organizers, Dr. Aisha Al-Dhahiri, Dr. Layla, and the whole team for this very well organized and interesting conference. Inshallah, we will meet you next year in person. Today, my talk is about the results of a study that was done from the MESC cohort. It's called Gestational Weight Gain and Gestational Diabetes among Emiratis and Arab women in the United Arab.
Dr. Hadi, there's something wrong with the sound. Your voice disappeared.
Is it here now? It's clear? Yes.
Yeah, it's fine.
Okay. So the results of the study of gestational weight gain and gestational diabetes among Emiratis will be presented in this talk. First of all, I would like to present the introduction is about, it's based on the hypothesis of the developmental origin of health and diseases hypothesis, which has found that the first 1,000 days of life between the woman's pregnancy and the baby's second birthday is a unique window of opportunity. And to grow healthier individuals. More and more emphasis has been placed on the role of nutrition in the prenatal and postnatal period. In this critical period, prevention should occur. Any prevention processes should occur during this critical, very critical period. So, maternal obesity and excessive gestational weight gain, maternal weight and weight gain during pregnancy has long been debated and to be one of the key markers of intrauterine nutritional environment due to their association with adverse obstetric and neonatal outcomes. Early life influences beginning in the intrauterine environment and continuing through the first few years of life shaped the trajectory of weight gain and body fatness throughout the life course. So it is important in this, let me remove this one. It is important to control the maternal weight gain and prevent excessive weight gain in order to lower the risk of adverse outcomes for mothers and their babies. So in 2009, the Institute of Medicine published revised gestational weight gain guidelines that are based on the pre-pregnancy body mass index. So as you can see here, the recommendations of the gestational weight gain during pregnancy is based on if the woman was, pregnant woman was underweight, her recommendation of gestational weight gain during pregnancy will be 12.5 to 18. But if she was obese, this recommendation will be much, much less. So in order to control the weight gain of the mother during pregnancy, why? It's important to monitor the weight gain during pregnancy, since improper weight gain can affect actually the health outcomes of the baby and the infant, of the mother, and there will be like adverse consequences, even if the mother gained insufficient gestational weight gain during the pregnancy. So she might be having more prevalence of preterm, low birth weight, and small for gestational age babies. As for there are also risk factors associated with excessive gestational weight gain, like the mother will be more, the prevalence of cesarean delivery is more among women who had higher or excessive gestational weight gain, and there was more chances of giving birth of a macrosemic baby, or increased, they will retain their postpartum weight retention after delivery is higher, and there was more prevalence of gestational diabetes. So, moreover, many studies have reported that GWG or gestational weight gain have been strongly associated with infant body weight outcomes. So we're not talking about mothers only, even infants are influenced with this gestational weight gain. Lau et al. In this systematic review have found that higher total gestational weight gain significantly increased the BMI Z-score of the infant and increased the risk of overweight and obesity of those infants from 1% till 2%. So, this is GWG is a potential risk factor to prevent childhood obesity. Also, higher maternal weight before pregnancy is reported to be associated with higher risk of gestational weight gain and increased risk of infant obesity. So not only gestational weight gain, even the mother's BMI before pregnancy is reported to influence infant obesity. So the negative consequences of coupling higher pre-BMI with GWG seem to reach beyond the period of pregnancy and contribute what we call transgenerational obesity. So it was concluded from many cohort studies that offsprings of obese mothers could inherit the genetic potential for obesity later in life. So what's the situation here in UAE? Actually, in UAE, as we all know, obesity is highly prevalent among mothers and infants. Many reports have reported that, showed that more than 50% of the women in the UAE were overweight or obese. So to best of our knowledge, there is little known about the patterns of GWG and pre-BMI and GDM in the UAE. So this study was aiming to investigate the patterns of GWG and GDM and the risk factors among cohort of Emirati women and Arab expatriates in UAE. This study was drawn from the MISC cohort. This MISC cohort was a prospective 2-year cohort study that included 256 pregnant women. They recruited the mother during their 3rd trimester from different prenatal clinics in hospitals in Dubai, Sharjah, and Ajman in UAE. Inclusion criteria was the mother has to have a singleton and she has to be pregnant with 3rd trimester, aged 19 to 40, from Emirati or Arabic expatriate citizen. And after delivery, we included only the mothers who delivered singleton. For the purpose of this study, we have collected that data upon recruitment was collected by face-to-face interview and by hospital records. We obtained some information from the hospital records, like the pre-pregnancy weight and the GDM diagnosis. From the mother, we collected so much. Multi-component questionnaire was used that included information from dietary intake, sociodemographic, lifestyle, and so many other data that is being analyzed more. This study was funded by Al Jalila Foundation, as well as a grant from the University of Sharjah. Also, this MISC was supported by Sharjah Baby Friendly Office and Medella. We got different ethical approvals from different research ethical bodies in UAE, from the Ministry of Health, Dubai Health Authority, University of Sharjah, Al-Qasimi Research Center. The variables, exposure variables, were the pre-BMI, pre-pregnancy BMI that was calculated and classified according to the WHO. The GDM was obtained from the clinical record, and the GWG was calculated as the difference between the recorded pre-pregnancy weight of the mother and the last weight measured before delivery. After that, we categorized the GWG according to IOM categories guidelines as having either insufficient weight gain or adequate or excessive weight gain relative to their BMI. Data was quoted and analyzed by using SPSS. So the results of those 256 mothers included that, reported that majority were from Arab non-Emirati origin, and the mean age was 30 years old, and the majority had high school diploma and technical degree. 19.1% of our sample had GDM, and this is considered a high percentage according to the world prevalence rate. For the maternal data, the mean pre-pregnancy BMI was 27.3. The majority had cesarean section delivery, and they were multiparous, meaning they had more than one child, and about 70% of them, they consumed their breakfast daily. As for the BMI, we noticed that, or as reported that, this is really worrisome, that the majority of, or 60% of the participants were either overweight or obese. And coupled with this, also the 41% of the mothers had gained excessive weight during pregnancy, and 23% gained insufficient weight during pregnancy. So this comes out to be 73% of the mothers had abnormal or improper gestational weight gain. And if we look at this figure, it shows the distribution of various categories of gestational weight gain as related or according to the pre-pregnancy weight. as you can see, it shows clearly here that underweight mothers gained insufficiently, the majority gained insufficient weight during pregnancy, while the overweight and the obese mothers gained excessive weight during pregnancy. So this means that our population did not follow the IOM guidelines. So if we look, so what did we do? We investigated the associations of various characteristics of the study participants with GWG using simple and multiple multinomial logistic regression. So the result showed that the result of the simple regression showed that parity and pre-BMI, pre-pregnancy BMI were significantly associated with GWG. These findings were confirmed by multiple multinomial logistic regression after adjustment of age. We found that multiparous women were 83% less likely to have excessive weight gain, and women with pre-pregnancy more than or equal to 25 had the risk or the odds of 2.23 times of gaining excessive weight. As for the association of the mothers with GDM, characteristics of the mother with GDM, the simple logistic regression showed that results showed that mother's age, consuming breakfast daily, and the BMI before pregnancy were significantly associated with higher odds of GDM. So after adjustment and the multiple regression showed that only women, the pre-pregnancy BMI showed that more than 25 BMI, more than 25 kilograms had 2.37 times the odds of having GDM. And as for the third, and we have noticed, as you have noticed, that pre-pregnancy BMI has emerged as a strong predictor for GWG and GDM. So we wanted to examine what were the characteristics associated with this pre-BMI of the mothers. So the results of the simple regression showed that mother's age, education, parity, and consuming daily breakfast were significantly associated with the pre-pregnancy BMI, but after adjustment and the multiple regression analysis showed that older women or women age and consuming breakfast were significantly the most significant factors associated with pre-BMI. So in conclusion, this study is the first study which prospectively investigated GWG, GDM, and its correlates. And this is the first one to use the international IOM in the UAE. What is alarming that has been reported by this study, that the high prevalence of excessive gestational weight gain and GDM among the participants. And what we found that, and this is in comparison with other studies, that pre-pregnancy BMI was found to be the common or the strong predictor of GWG and GDM. So based on these, overweight and obese women should be given special attention by the healthcare specialist during their visit, clinic visit. The healthcare specialist shouldn't focus only on the fetus health. They should focus also and give the high-risk women who are overweight and obese more attention during this visit, and advise them, giving them specific advice on how to gain proper weight gain during pregnancy, and how to follow and advise them to follow healthy, balanced diet, and the importance of good nutritious breakfast, as well as increasing the physical activity. Finally, this study highlights the importance of developing strategies aiming at preventing obesity. By preventing obesity in young women, we are actually will be preventing the progress or the prevalence of GDM, excessive gestational weight gain, and decrease the obesity-related factors in subsequent pregnancies. This study would not be done without the great MISC research team. I would like to thank my colleagues, Professor Riyad Ubaid, Dr. Haidar Hassan, Dr. Muna Hashim, Dr. Farah Naja, and Dr. Hassan Ghazal, and special and deep thank to our dedicated research assistants and all the staff and the nurses who worked in these different sites in the Sharjah, Ajman, and Dubai. And thank you all for listening.
Thank you so much, Dr. Hadia, for this nice, interesting presentation. And thanks for adhering to the time as well.
Yeah,
I could see some questions, but I prefer to keep it till the end of the session. So I would kindly ask the presenters to stay with us till the end of the session today. And with this, I could move to the third presenter of today. Again, my colleague, Dr. Mu'azz al-Islam Faris. Dr. Mu'azz, he got his 3 higher university degrees from the University of Jordan in the field of human nutrition. And he has recently completed the Harvard Medical School Global Clinical Score Research Training Program. He is an associate professor at the Department of Clinical Nutrition and Dietetics at the University of Sharjah. He published over 100 scientific publications, 3 books and chapters. And today's presentation, he's going to talk to us about intermittent fasting, the caloric restriction, and healthy aging, what the science says. So please welcome with me, Dr. Mu'azz al-Islam.
Assalamu alaikum wa barakatuh. Thank you so much, Ms. Sundus, for this nice introduction. First of all, I would like to thank the organizers and the chair of this conference, Dr. Aisha Dhahri, and the organizing and scientific committees, with special thanks for Dr. Layla and other colleagues and members. My talk, I think this is the last session, our last talk, صح, Ms. Sundus, this is the last one. إن شاء الله يكون مسك الختام إن شاء الله. It is about caloric restriction, intermittent fasting, and healthy aging. First of all, on the behalf of myself and my co-authors, I declare that there is no actual or potential conflict of interest in relation to this presentation. I would like to start with the Holy Quran, as Allah Azza wa Jal mentioned in the Holy Quran in 2 positions indicated for the elderly people and for the aging stage of the human life, and grasps our attention to the critical period the human being is passing through in the elderly. Allah Azza says, وَمَنْ نُعَمِّرْهُ نُنَكِّسُهُ فِي الْخَلْقِ أَفَلَا يَعْقِلُونَ, indicating that drastic changes in the human body and the degenerative and deterioration consequences that occur during this critical stage of human life. And the other ayah, قال الله عز وجل قال رب إني وهن العظم مني واشتعل الرأس شيبة على لسان داود عليه السلام ولم أكن بدعائك ربي شقيا. All this will give us attention about the elderly people and how much we have to take care about these people. So in this outline, I will talk about the definition of aging and its types, the main physiological changes during aging, the role of caloric restriction and intermittent fasting in human health and longevity, and the impact of caloric restriction and other types of intermittent fasting in the new degenerative diseases, and the mechanisms how caloric restriction, periodic fasting, and intermittent fasting impact the brain health and the neuroplasticity, and the Islamic model of Ramadan during intermittent fasting, and its expected implications on the brain health and neuroplasticity, and again, finally, the conclusion. So first of all, the definition of aging. Aging is actually defined as a physiological process that results in progressive decline in multiple organ systems that's affecting the reproductive, metabolic, physical, and cognitive functions, as well as affecting the human survival. There are two types of aging. The first type is called the primary aging, or the intrinsic senescence, which is the progressive deterioration in physical structure and biological function that occurs with advancing age alone or independent of other factors. So these are the intrinsic, the inherent changes that occur during the aging process, regardless of the presence or the absence of any other triggering factors like the chronic diseases. These intrinsic senescence or primary aging is associated with decrease in the bone mineral density, decreased muscle mass, abdominal fat accumulation, the progressive decline of cardiac, pulmonary, renal, and immune function that occurs normally with increasing the age. The second type of aging is the secondary aging, which is the accelerated deterioration in organ structure and function that is mediated by the chronic disease that the elderly people mostly suffering from, like diabetes and hypertension, or the exposure for other environmental factors that accelerate the degenerative processes and the deterioration of the body organs and systems, like smoking and the excessive sun exposure, and in other cases, alcohol consumption. So what are the main physiological changes that occur during aging? Maybe the oncologist or those who are interested with research on cancer, they know this hallmark of cancer image, or this representative image. The same thing the scientists developed for aging, as aging is characterized by a set of unique changes at the cellular, genetic, metabolic, and the whole body functions. For these 9 features that characterize the aging stage of human life include the genetic susceptibility instability, which increases the risk of having DNA damage and increasing the risk of cancer. So this is why the age is one of the risk factors of developing the human cancers. Telomere attrition, as you know, the telomere looks like the cap on the thread that covers that thread from any kind of damage. But once this telomere is decreased, this will increase the risk of having different types of changes, deteriorative changes and damages that may affect this chromosome. Also, there is a kind of genetic alterations and epigenetic alterations, because the genes are fixed. So the epigenetic alterations like the acetylation, histomodification, and other epigenetic processes, also they are changed in a way that also increase the risk of having chronic diseases, especially cancer, like in the acetylation or lack of normal acetylation of the genes, which is associated with the risk of having colon cancer. The loss of proteostasis, which is the homeostasis of the protein synthesis at the cell level and surrounding the cell. And this is directly reflected on the abnormal regulation of many, or lack of normal regulation of many of the basic metabolic processes due to the lack of the proteomic expression and the proteomic changes that happen working normally in the normal stage of life. These proteomic expressions and protein regulations are disturbed, so this will lead a kind of metabolic consequences that affect the human life. The deregulated nutrient sensing and the lack of normal sensation, which adversely affects the ability to taste the food, and so will be affecting the total caloric intake and the total macro and micronutrient intakes, which in turn will affect the nutritional status and lead to some types of micronutrient deficiency, or maybe, though at the worst case, the protein energy malnutrition with the deteriorative effects of this kind of malnutrition. Also, the mitochondrial dysfunction and the inability of the mitochondria to do what's called the biogenesis of the energy, which fuels the body and helps the body to maintain and to do the normal exercises and the routine life exercises. All these will be adversely affected by this kind of mitochondrial dysfunction that characterizes the elderly stage. And the cellular senescence, this makes the cells like a kind of lack of function cells without any clear function in the body, just a kind of garbage cells that the body has to get rid and to destroy. But these senescent cells will lead to other problems if not removed by some mechanisms activated in the body, like the autophagy and other mechanisms. Stem cell exhaustion, and finally the altered intercellular communication. All these, the 9 unique characteristics or hallmarks of the aging process. Looking at the mechanisms that involved in these deteriorative and degenerative processes that are characterizing the elderly stage, we can find that the first thing is the oxidative stress and the increased risk of having oxidative stress, which leads to induced protein and DNA damage and genetic instability of the mitochondria and the nuclear genomes. The other dysfunction is the non-infectious chronic inflammation, which is characterized by elevated pro-inflammatory cytokines like interleukin-1 beta, interleukin-6, and TNF-alpha, which also exaggerate the problems and the risk of having chronic diseases and increase the severity of the chronic diseases as a result of this elevated inflammation, which is not related to any kind of infection, because this is a kind of a single identity of inflammatory process that is not related to any kind of infection. This is inherited inflammation associated with the aging process. The alteration in the fatty acid metabolism, which is associated with excessive fatty acid released into the plasma. This will lead to an increased risk of having insulin resistance that characterizes the elderly people. The accumulation of the cellular garbage, as just we mentioned, and this leads to the accumulation of the advanced glycation in the products, amyloid, and the proteins that interfere with normal cell function, and this will increase the risk of having these neurodegenerative diseases like Alzheimer and other neurological diseases. And finally, the alterations in the neuroendocrine system, which affects the sympathetic nervous system and the angiotensin system activation, and all these will lead at the end to a list of symptoms that characterize the elderly phenotypes, including the sarcopenia, frailty, immobility, the mild cognitive impairment, and the incontinence, and other chronic diseases which are associated with the elderly age, like dementia, cancers, atherosclerosis, diabetes, osteoporosis, osteoarthritis, renal dysfunction, blindness, chronic lung disease, and other symptoms associated with decreased resilience, like increased risk of infection, delirium, delayed wound healing, slow rehabilitation, chemotherapy toxicity, and the ICU admission. Looking at the inflammatory markers and immunological changes that are associated with the elderly, we can find clearly that we have a set of inflammatory markers that characterizes this age, this stage of elderly. These, like the interleukin-6 and the T and TNF-alpha, which are highly associated among the elderly people and responsible for many of the degenerative problems and diseases that associate with this stage of life. Decreased the anti-inflammatory cytokine, which is the interleukin-10, which characterizes this stage also, and the gut dysbiosis, which is associated with a lack of normal balance in the microbial quantity and quality among these people. Also, there is a kind of dysregulation in the immune system as revealed in the innate immune system by increased neutrophil level, monocytes, and natural killer cells, and in the adaptive or acquired immune system associated with decreased naive T and beta cells, memory T cells also increased, and the exhausted T cells also will be increased. At the end, all these immunological and inflammatory changes will be resulting in a kind of deterioration in many of the body functions and body cells, including the bone marrow, as presented in decreased hematopoietic stem cell self-renewal and the hematopoietic stem cell lymph node differentiations, and decreased pre-beta cells associated also with increased inflammatory cytokines, decreased interleukin 7, which characterizes the normal people, and the thymus gland functioning, and increased adiposity, which is also one of the major risk factors that increase the oxidative stress and inflammation among these elderly people. There is also another, which is called another deterioration, which is called SASP. We'll discuss it next slide, which is characterized also this stage of life for the elderly people. And this stage is characterized by increased interleukin 1 alpha and beta interleukins, increased interleukin 6 and 8, and increased TGF beta and CXL, which is also all chemokines associated with increased risk of inflammation. So although we have these unique characteristics that affects the human life at this aging period, but however, there are a list of interventions that can, to some extent, modify the severity of these deteriorative changes and alleviate the complications of aging on the human health. Yes, we know, we believe our life is fixed, something can't be changed, but the science says that there are some factors may help to alleviate the deterioration that is associated with the elderly. And other studies, they found that those people who have healthy interventions, they got healthier life, and others, they got a longer lifespan, which indicates that there could be a kind of a major influence for the environmental factors in changing these deteriorative effects of the aging process. Yes, the genetics are fixed, but as I mentioned, the epigenetic processes may alleviate these problems. Some drugs that affects the longevity and the neurodegeneration also could help in lowering these symptoms. Some metabolites like the beta-hydroxybutyrate or the part of the ketone bodies that have been shown to have a positive effect on the neuroplasticity and improve the brain health. The diet with the list of the antioxidant, anti-inflammatory compounds and the factors that affects the epigenetic modifications in a way that help to suppress the deteriorative changes associated with elderly, and exercise. All these factors have been shown to affect the elderly in a positive way, decreasing the severity of loss in the bone, the cartilage, the muscle mass, and the strength, decrease the gain in the abdominal fat, which characterizes the elderly people, improves the altered hormone levels, improve the mechanical and structural changes for the elderly people, decrease the risk of multi-morbidity, and decrease the risk of frailty among the elderly. So human senescence and cell senescence, one of the features that characterizes the human aging, this is presented in what's called the SASP, which is known as the senescence-associated secretory phenotype. There is a set of phenotype characteristics that characterize this stage of life. And these senescence cells have been implicated in a number of biological functions that ranges from the or stages from the embryogenesis until the aging process. Also, the research found that caloric restriction and intermittent fasting are effective non-genetic interventions that help to delay these genotypes associated with the SASP. And caloric restriction and intermittent fasting showed in many evidences in the human and in the animals that this intervention, one of the best and the most effective and safe interventions that helps to decrease the risk of having aging and the associated deteriorations. So how the fact the intermittent fasting and caloric restriction affects. I always start with this scientist, Professor Osomi, the Nobel Prize laureate in the 2016, who got his Nobel Prize in medicine as a reward for his efforts in discovering the autophagy, which he found that this autophagy mechanism, which is integral in fighting and alleviating the the degenerative and deteriorative processes associated with aging was triggered actually by fasting, and especially the intermittent fasting pattern, as we can see here. So, Professor, also, yeah, please.
Well, allow me just to 3 minutes left over.
3 minutes.
Yeah.
Are you sure?
Sorry. I'm sure.
Okay. طيب. So I will try to jump سريعا على الميجر أو المين review articles that shows how the fasting help to improve the senescence and alleviate the aging by removing the immunosenescence and killing the old and damaged cells and replacing these cells with more functional cells. Fasting has now many clinical implications, including the alleviation of many of the symptoms associated with aging, especially those associated with increasing the brain-derived neutrophilic factor, the decreased levels of the inflammatory markers, TNF-alpha, interleukin-1 beta, interleukin-6. There are a list of changes, physiological changes that characterize this period of aging, including the decreased IGF-1, because I want to share our results about the IGF-1 and how these IGF-1 are associated with how the reduction in the IGF-1 associated with decreased level of neurodegenerative diseases. So dietary restriction and caloric restriction have been shown to improve the brain volume, dendritic spines, gray matter, to improve the neurotransmitters, increasing the brain-derived neurotrophic factor, decreasing the reactive oxygen species, and so it helps in improving the neurogenesis, neuroplasticity, and improves the neuroprotection and improves the cognitive function at the end. This one study showed how the alternate day fasting, which is called David or Dawood alayhi salam fasting, helps to decrease the anxiety and improve the quality of life. And this reflects to a kind of improvement in the central nervous system function, because the stress and the anxiety are reflect for a kind of mental stress that the human being exposed during the life period. So fasting has been proposed as one of the best therapies for the neurological disorders associated with aging. How? By increasing the beta-hydroxybutyrate, brain-derived neutrophilic factor, adiponectin, ghrelin, and decreasing the other factors that have been associated with increased inflammation, like the interleukin-6, TNF-alpha, the mTOR, which is decreased, and so the autophagy is activated in the body. All these factors actually help at the end to optimize the nervous system and decrease the severity of aging. This study shows how the regular practice of intermittent fasting help to decrease the DNA damage, help to increase the mTOR, which is the autophagy marker, decrease the mTOR, this one. Decrease the C-reactive protein, increasing the SOD2, اللي هو superoxide dismutase, one of the major antioxidant enzymes, and decreasing the malondial dehydrogenase, one of the most important indicators for the oxidative stress. Brain-derived neutrophilic factor have been shown to be increased, along with other genes of oxidative stress have been shown to be increased with the practice of intermittent fasting and time-restricted feeding. IGF-1 have been shown to be decreased actually with the practice of the intermittent fasting, like time-restricted feeding, and BDNF have been shown to be increased also with the practice of intermittent fasting. So IGF-1, one of the factors that are associated with aging and elderly people, and a meta-analysis shown that the practice of fasting and intermittent fasting have been shown to decrease the IGF-1, but this helped to lower the risk of having neurodegenerative diseases. In our study, we found that the subcutaneous fat and the visceral fat also improved, and the visceral adiposity decreased during fasting in Ramadan, which helps at the end in alleviating the pro-inflammatory cytokines that are associated with the elderly stage, like the interleukin-6, TNF-alpha, IGF-1, which I just mentioned, and increasing the anti-inflammatory cytokine like interleukin-10, which has a neuroprotective effect and anti-inflammatory effect. this study about the genes that regulating the enzymes, antioxidant enzymes, and the inflammatory mechanisms showed that fasting in Ramadan as a model of intermittent fasting has a significant improvement in the NRF2, which is a master gene that helps to regulate the antioxidant enzymes and the antioxidant capacity in our bodies. But this help in alleviating the oxidative stress and the associated inflammation. The same thing for the TFAM, which is responsible for the mitochondrial biogenesis, which means that fasting as a model in Ramadan helps to activate the mitochondrial functioning and alleviate the mitochondrial deterioration, which characterizes the elderly stage. SOD2, which is a major also enzyme in the antioxidant system, also increased significantly at the end of Ramadan, implying that the practice of 1 month of Ramadan fasting as a model of intermittent fasting is helpful in alleviating the background conditions associated with aging. Third one was not significantly increased, but 33 was significantly decreased, which could be explained by the lack of significant changes actually during Ramadan in the caloric intake. This does not lead to any increased expression of this hormone and the protein of that this gene and the protein of that gene, because this gene actually secreted once there is a kind of severe caloric restriction in our bodies. So these three major actually proteins, the NRF2, TFAM, and the SOD2 have been shown to be associated actually with the decreased oxidative stress, enhanced bioenergetics, decreasing the cellular stress and increasing the cellular resistance, and improving the mitochondrial biogenesis, and at the end improve the neuroplasticity of the human brains. I will stop here because the time is out, and will just conclude that for healthy aging, we have to take care about the diet of our elderly people. Caffeine has been shown to improve the central nervous system functioning, and caloric restriction and intermittent fasting, which is our title in this presentation, has a major role actually in alleviating the background changes associated with the aging and the deterioration and degenerative changes associated with the elderly stage, along with the exercise. All these factors will help to improve the elderly and people and decrease the chance of having kinds of deteriorations. وكما قال ربنا عز وجل وأن تصوموا خير لكم إن كنتم تعلمون. Fasting is something good that we have to search to know how fasting is good for our health. And at the end, اللهم بلغنا رمضان. جزاكم الله خير. Thank you so much for your kind listening.
شكرا دكتور معز على المحاضرة. Thank you so much, Dr. Muaz, for this interesting, lovely presentation. I liked it.
Thank you.
First, I would like to announce to the attendees, please stay with us, although it's the last session, but there will be two more workshops. And I think I have, just couple of minutes for a few questions, and I'm sure the presenters they will respond to your questions through emails. But first of all, is Professor Liliana with us? Professor Liliana. I liked your topic. I liked your topic, by the way. Probably I will send you a private question, but we don't have too much time for my questions. So I think we have here one question for you, Professor. Multiple questions. I think you responded to all the questions and inquiries, Professor Liliana. But I think here we have one question for you, Prof. Liliana. In milligrams, how much or how many milligrams we can prescribe for patients? In milligrams, how many milligrams of isoflavonoid we can recommend to post-menopause women? Can we prescribe?
Thank you for the question.
Milligrams.
Thank you for the question. It's we do not prescribe the isoflavones as such. Isoflavones are part of the phytoestrogens that are found in compounds. So basically for healthy aging, I strongly believe that incorporating in the normal diet, so phytoestrogens are important to be incorporated as normal diet, not as supplements, because we just don't know whether they are contaminated or whether the dosage is correct. So even though they are found in complementary alternative therapy, it's best to consume soy natural soy or soy milk that's got some calcium added to it, or most of the grains from soy down to beans, down to mainly fermented foods. So the Japanese food and the Korean foods are extremely important because of fermentation, the bifidobacterium in there. So that can stimulate the production from soy into diazoxide, from diazoxide into equiol. So we do not prescribe as such what you said, but consuming it via food.
Amazing. Thank you, Professor. Thank you so much. I'll get back to you for some questions for sure. For Dr. Muaz, I could see many questions, by the way, Dr. Muaz. Your topic is interesting as well. But I select just one question. I'm biased with this question only. How about intermittent fasting and neurological disorders such as multiple sclerosis? Is there any relations, or has been any kind of studies?
Yes, thank you, Dr. Sundus, and thank you for the speaker, for the audience for this question. Actually, yes, there are some research about the impact of intermittent fasting against the autoimmune diseases and inflammatory diseases that affects the human body, including also the multiple sclerosis, because multiple sclerosis is kind of neuroinflammation, and with the neuroinflammation, the severity of the disease is increased. And as we just presented in our study and other studies, that the practice of intermittent fasting helps to alleviate the pro-inflammatory cytokines and other inflammatory markers. So this kind of alleviation is expected to have a positive effect, actually. And there are some research about the effect of fasting in general and intermittent fasting in particular in alleviating the multiple sclerosis. Yes.
Thank you, Dr. Muaz. Didn't find any question to Dr. Hadia, but if there's any question right now, anybody could have any question to Dr. Hadia? Any question to Dr. Hadia? For all the other, attendees, I'm sure the presenters will respond to your questions through email. I have a very short question, Professor Liliana, if you can just, a few words. According to your study, I think you are recommending the Korean or, sort of Asian diets. My question about your study, what about, recruited participants? They were all, from all nationalities, or they were all, Asian, Korean? From which nationality did you recruit your group study?
Thank you for the question. I presented 2 studies of ours. Both are said they're pilot because there were like 40 people in each. But the one on the supplements with Maca, which is available, as I said, as a powder, even in UAE and throughout the world, we recruited in Australia Caucasian women, and then I spent 6 months in Hong Kong. We recruited Asian women. We really wanted to see the difference between Caucasian and non-Caucasian, because Asian women do not complain and they do not have severe symptoms. However, they have osteoporosis, a lot more than Caucasian or other type of women. So basically their lifestyle, when I say soy, everyday soy, and other Asian women from Japan and Korea, they have miso. So their lifestyle is from birth, that is from young age to old age. While we, women of other nationalities and culture, tend to have that at the time of menopause, thinking we're going to combat menopause and symptoms with only if we have diet now, but it doesn't last. The body really needs to have a long-term effect of some of these nutrients and also microorganisms in the gut.
I do agree with you. Like when I talk about bifidobacterium or, probiotics, it should be every day. Thank you so much again. Thank you so much.
No, it should be every day. But what I was saying, we are not producing, the other women non-Asian are not producing equiol. And equiol is the active compound that has a very similar chemical structure like estrogen. So why 70% are non-producers of the Caucasian and other women, and most of the Asian are equal producers? We are basing it predominantly of their dietary intake throughout the life of soy.
Amazing how nutrients change our life. Thank you so much, dear presenters. And now I think we have to move to the second workshops. Thank you so much.
Thank you so much,
Ms. Sundus, for your organization. Thank you.
Liliana
Before you leave,
Gomez.
can we
Thank you
take a
all.
group photo, please? Can you all look at your laptop camera in a count of 3? Okay, in 1, 2, 3, smile. Okay, thank you so much for your time, Ms. Indus,
Thank
Professor.
you. Thank you all.
Thank you
Thank you
all.
so
Thank
much.
you very much. Thank you, Kim. Thank you.
All right, attendees, thank you so much for your time all. Please stay tuned with us. We still have an upcoming 2 workshops at exactly 6 o'clock. Please visit our exhibition booth with the link that we have provided with you. All the sponsored companies, Nestle, Tuat, Abbott, and Sika Nutrition Society, Emirates Nutrition Association have their own booth. And once you click on them, it will direct you to their personal booth with a chat functionality, which a representative will be able to answer for any of your inquiries or clarifications. We also have the posters inside the exhibition. Once you click on the poster wall, it will direct you to all the oral presenters that we have. So please visit our exhibition hall and our abstract hall. And we look forward to see you again at exactly 6 o'clock UAE time. Thank you. Hi, Mr. Mark. Hi, Mr. Mark. Mr. Mark, can you hear me? Mr. Mark? Attendees, we will just pause for a short break. Please stay tuned on the Zoom line. I will reconnect the Zoom at exactly 5 minutes before 6 o'clock. Thank you, and please come back again with the same link.